alanine, also known as ALA or A, is one of the 20 amino acids that are within the human body that make proteins. All of the amino acids have a carboxylic acid where you've got 2 oxygens, a hydrogen, and a carbon. You've got this chiral carbon, the central carbon, where everything's attached to, a nitrogen in blue with 3 hydrogens when it's fully protonated. You've also got another hydrogen that's on the back of this chiral carbon. That's always going to be away from you. You're not really going to see it if you're making a model. And then every single amino acid is going to have some different type of R group. As it relates to alanine, this R group is a CH3 or a methyl group. So we've got carbon with 3 hydrogens attached to it. This is the L form of alanine, the L coming from how it looks like in relationship to glyceraldehyde, and so this is L. There's also a D version. Different places you're going to find the D-alanine versus the L-alanine. To get started, let's just talk about the L-alanine. So if we're talking about the amino acids, we've talked about the carboxylic, we've got the nitrogenous, but as it relates to alanine, all that really matters is the R group. It's R group, methyl group. We've got 1 carbon, 3 hydrogens. Every single one of these bonds for this carbon to these hydrogens is going to have an equal sharing of electrons, aka not polar. It's going to be strictly, purely covalent. There's no unequal sharing. And because there's no unequal sharing of the electrons within this R group, this is considered a non-polar amino acid. And because it's non-polar, it's not able to do any hydrogen bonding. That's where they take advantage of the fact that there's an unequal sharing of electrons. None of that's occurring. So this is also going to be hydrophobic or a water-fearing. So alanine is both non-polar and hydrophobic as an amino acid because its R group is equally sharing all of its electrons. Now, with all amino acids, we can talk about something called the pKa, and that's the pH, or the amount of hydrogen at which the hydrogens are going to be lost from this molecule. If you have a non-polar molecule, this methyl group isn't losing any hydrogens, so it's irrelevant here. We're going to have a pK1 and a pK2. Carboxylic acid is the acid, and acids by definition give up hydrogens or accept electrons. In this case, it's going to lose its hydrogen here. At a pH around 2 something, the hydrogen that was on the COOH is now gone, and now it's a COO minus. Now, this nitrogen should only have 3 bonds, and it actually has 4 right now. And so because it has an extra bond, this nitrogen is carrying a +1 charge. This oxygen is going to have a -1 charge. So if we have a +1 and a -1 on the same molecule, +1 and -1 add up to 0. But the actual molecule is not 0. There are spaces where it's positive and negative charge. This, when you have a positive and a negative charge on the same molecule, but it equals out to 0, is called a zwitterion. And so at the physiologic pH, or about 7.4, which is how the pH of your body is most of the time, this is what alanine is going to look like. The hydrogen's gone from this carboxylic acid, but this nitrogen still has an extra hydrogen. This nitrogen isn't going to lose its extra hydrogen until a pH of something like 9. And your body's never going to be at a pH of 9 and still be functional. So at all points in time, the amino acid that you're going to see actually within the human body looks like this. We've got NH3 plus and this COO minus because you have 1 fewer bond on this 1 extra oxygen. This amino acid is not considered an essential amino acid because you can make it within the human body. There's a lot of different pathways that are used to be able to make alanine, but most of them require some type of transamination, or where you're moving this amine group from one thing to another thing. So transfer, movement of the amino group amine. And then nation always just means like the action of doing something. So transamination is how you end up getting alanine. And because you can transaminate a molecule to get alanine, it's not going to be essential. One of the main enzymes that is involved in the transamination to make alanine is called alanine aminotransferase. The initials are ALT, and this is one of the most important enzymes that allows for alanine to be created. The majority of this enzyme is found in your liver. And so if you have elevated ALT or your alanine aminotransferases, there's too many of them within the liver. Some of that's going to bleed out into your serum levels or into your blood. And so you can check to see if your serum or the levels of ALT in your blood are normal or elevated. If those numbers are elevated, that means your body is making way too much ALT for some reason. We don't know if that's because the liver is malfunctioning or something else is malfunctioning that's making the liver work in overdrive, but you can do follow-up tests to figure out, is something else causing the liver to malfunction or is the liver malfunctioning all on its own? But either way, ALT, the alanine aminotransferase, is one of the enzymes that is in your liver that you test in your blood to see if your liver is functioning too much. Now, you remember how I was talking about the L form of this amino acid? You also can have a D form of the amino acid where the nitrogen and the hydrogen switch spots. The nitrogen's now pointing up. And because you've got the molecule looking like this is the D form of this amino acid. This isn't about to make any proteins at all. But it is going to have a major role within the peptidoglycan layer of bacterial cell walls. So if you look at a peptidoglycan layer, you'll notice that there are a whole bunch of little groups of glucose or some other sugar that stack together. And then each of those polysaccharides, the sugars molecules joined together, is going to be cross-bridged with some kind of a peptide. That oligopeptide or a small peptide is mostly made of alanine. And it's not the L-alanine, it's D-alanine. If you're trying to create a drug that's targeting only something bacteria have that humans don't have, one of the things that drug can target is D-alanine. Your body is not about to use D-alanine, your body's not about to make D-alanine, but that bacteria that's infecting your body is going to have D-alanine. And so if that molecule is just targeting the D-alanine, it can kill off the bacteria and leave you alone.